It's amazing that explaining life's immense diversity all comes down to some genetics and some biochemistry. And life on Earth is just one family. And what's true for you is true for all biology.
Hello, welcome to Genetic Shambles. It's Wednesday. It was 2 weeks ago since the last one. That's why we're doing it again now. We do these fortnightly. And this is a show where we'll be talking with people who, unlike many of the people you've been seeing on television talking about pathogens recently, these people actually know what they're talking. They've spent a long time studying them. They kind of know things. They're not people who've just been dragged off a street to, cut up a mask for some kind of TV stunt or whatever. So this is, I should say, Genetic Shambles. That's very specific to something that happened this week, but I won't go on any form of soapbox. Just join me when I'm drunk on Facebook, and I'll continue this. Anyway, Shambles is presented in association with the Genetics Society and the Milner Centre for Evolution at the University of Bath. And all of the episodes we've done so far are available up on video form and also in audio form as well. So you can go back. All of these are recorded, and we've talked about lots of different things. Obviously, we started off, unsurprisingly, doing panel all about COVID-19, but we've been talking about the nature of evolution, our understanding of genetics from the perspective of species divide, and we might even eventually, I don't think we've done yet one about behaviour, which of course is such an interesting area and a hotly debated area, but we might get round to that. So that one doesn't exist as yet. All the others do, though that one does also exist in the Nature of the Block universe, but it's inaccessible to you for the time being. Also, I should mention the Genetics Unzipped podcast. That is another place you can find us, but you can also find Kat Arney, who was on 2 weeks ago as well. And so you can also go to YouTube for the live chat. And if you have got any questions during this show, which I said is specifically about pathogens, but may well be tangential at times, then we will ask live questions as well. So if there's anything you'd like to ask, I'll be keeping an eye on that. Now, our guest today, the first someone who I last saw out in New Zealand when we were doing gigs in Wellington, Auckland and Christchurch, and I saw her in Wellington and Christchurch, Dr. Siouxsie Wiles, who is a microbiologist and associate professor at the University of Auckland, and has also been one of the leading voices of scientific information in the fight against COVID-19 in New Zealand, where, of course, it has been perhaps somewhat more effective. In fact, we may well go off on a tangent very early with this, where we're broadcasting from, who could really do with some good advice. We're also joined by Jenny Rohn, who's a principal research fellow at University College London, and sorry, I did that. University College London, very much in a university challenge way. There are no questions. Research bacteria and their human hosts, which is why she has a slightly more ghostly light. You will actually see their introduction, bacteria and their human hosts, behind her on the bookshelf. Many stories, great stories of bacteria. And we also have Lavanya Mane as well, final year PhD student in the tuberculosis laboratory at UCL and the Francis Crick Institute, where she is researching the metabolic interactions between prominent respiratory bacterial pathogens. So, well, actually, let's start. I said we'd go off on a tangent, and so we'll start on a tangent if you can. Siouxsie, just because you have been, what have you found have been, from the perspective of your own discipline, some of the misunderstandings that you've seen during the COVID-19 crisis, and some of the things that you have felt the need to correct, which perhaps been in the mass media?
Oh, where do we start? Oh my goodness. Gosh, I think one of the things I've really learned is also how much of our knowledge was perhaps based on dogma or really old experiments. One of the problems I think with infectious diseases as a research area is they've been quite neglected really for a long time, right? Lots of the money now is pushed into cancer and various things, and so there's stuff that we we know, especially around transmission of infections, that's actually based on some quite old experiments. And so, you know, there's been arguments about sort of the technical meanings of words like airborne and aerosol and things that have, you know, very specific meanings to microbiologists and infectious diseases people. But it kind of ended up being almost like, and asymptomatic, there's another one, asymptomatic and pre-symptomatic, which became almost like technical arguments that started to really muddy the water about people's understanding of what they needed to do. So that's sort of been really interesting. The other thing I think that's been really fascinating has been how there's this huge focus on the virus. And very little real understanding that it's just part of what we call the disease triangle that involves also the host and the environment. And what we're seeing around the world is just how important the environment is. And by that, I mean the political environment, you know, all of these things that can impact and human behavior, social behavior, how that can impact on this virus. So, yeah, what I've been kind of doing is just trying to keep an eye out on what sort of how the media is picking up on things and then going, oh yeah, no, there's something that people don't understand, and then trying to push out information that sort of tries to address that and tries to explain to people that it is actually quite complicated. And while we're trying to make things really simple, sometimes if you don't accept that it's complicated, people then just sort of seem to think either everybody's misleading them. And the other big thing, I think, is also how quickly things are changing and getting people to understand that our job as scientists is to keep looking at what evidence is appearing and changing our minds as the evidence changes. And so I've had so many people, you know, criticizing me saying, but you said something different in January. It's like, well, in January, we knew like almost nothing. And so this has also been, I think, quite a good opportunity to explain to people how science works. But we're trying to do that in just such a noisy environment. And I think that's quite hard, very clearly quite hard.
I wonder whether I'd like to ask you, Jenny, and Lavanya as well, perhaps starting with you, Jenny, are there certain things within this kind of pandemic that you would particularly like to underline in terms of, as you were saying, Siouxsie, this is the trouble is the information changes. This is something that is new that is being dealt with. This is something, and we are in a society where, as you mentioned before, many things are very dogmatic, and it's seen as being a flip-flop position, whereas of course science is a flip-flop position because the information changes. So, Jenny, is there anything you particularly found that you would like to highlight to people?
Yes, I think the infamous mask U-turn is a really big one. So in the beginning, the WHO and most people were saying, well, masks won't help. And it's true that your average common or garden cloth mask is not going to prevent you catching COVID-19 if you're confronted with a sneeze. But as time went on, it became clear that actually wearing a mask will help a little bit, and it's certainly a good idea. And now it's sort of everybody appreciates the idea that a mask is a good idea. But there, I'm on record back in March saying, "Oh no, don't wear a mask," and now I'm on record saying that you should wear a mask. And I find it a little bit uncomfortable because I know why I changed my mind is because the scientific sort of thinking changed, but people who see me and don't realize, as Siouxsie so eloquently put it, that science is a rugby scrum going back and forth. It's not just truth, it's this back and forth thing, and everybody's fighting about it. Even scientists are fighting. On Twitter today, I've been following with interest the leading epidemiologists sort of disagreeing with each other about the trajectory, like about what our famous UK 3,000 case spike, our spike means. It's sort of interesting to see scientists fighting in public and the public not understanding that this is normal.
Can I just ask, what is the change in terms of the understanding of masks?
Well, basically, I think masks were always a good idea, but there weren't. So especially in the UK, we couldn't advocate everybody wearing masks if people couldn't actually buy them because we didn't even have enough for our own workers. But I think, I don't know, maybe you others have an opinion on this. I think basically we wanted to prevent it 100%. And now we realize every little helps, right? Even a little bit of reduction in transmission will help. And a mask is a very easy thing that people can do. And it won't prevent all infections, but it will dampen them down. So I think we kind of had this perfectionist idea of masks, that they weren't perfect, so we shouldn't wear them. And that's not that's not tenable, really. I don't know what you guys think about that.
Yeah.
Lavanya?
So
sorry, Siouxsie.
one of the issues I think is this difference between whether people are wearing them to stop transmission, as in if they have the virus, or whether they're wearing them to protect themselves. And it's very clear that if you are wearing a cloth mask or a surgical mask, and at the time as well, we were much more concerned about fomic transmission. So this is the transmission of the virus from surfaces. So it's very clear if you have a mask that doesn't fit properly to your face, like tightly to your face, and you are not used to wearing one, and you are touching your face a lot and adjusting it, that is not a good idea if you think you're wearing the mask to protect yourself from becoming infected. And as Jenny says, there was a huge shortage everywhere, and the real concern was that people who were treating infectious patients and needed that as part of their full protective gear needed them. Now, of course, we know that fomic transmission, while it happens, is probably not a big contributor, and that actually it is much more about preventing you as an infectious person from shedding all of your virus. That changes everything, right? And people don't see that. They don't see the things that we were thinking, or what, even though we were trying to explain them at the time, they just see that, oh, you said they wouldn't work, and now you say they do. And it's because we were thinking about them as two very different things. And when you're in a place with very little transmission of the virus, then actually, you could argue that, well, actually, you should save them for the people who have, who need them. But now there's so much transmission everywhere. The chance that you are one of those infected people and don't know it is why everyone should be wearing a mask.
Yeah, and just to also say about the masks, the idea of you wear a mask because you are being considerate towards other people. By you taking this very simple action, you're trying to protect everybody around you in the community, and to really highlight that and show that that's still true, even though it may feel like we're getting kind of used to this new normal, but that doesn't mean that you should be complacent. I think that that's also a thing to really highlight, that you are protecting not just yourself, but literally everybody that you interact with. Yeah.
Brilliant. Thank you. I just wanted to have a bit, just because it does seem to be very much, debated very often with people who don't have the resources or understanding that you have. So let's start now with the broad theme, which is pathogens. And as usual, we will start with definitions. I know, Siouxsie, it's something you talked about in your book, which is the annoyance, the confusion by people like me between viruses and bacteria and what that means. So let's just start off basically with the definition of what a pathogen is.
Well, so pathogen is basically a microorganism that can cause disease, and that could be cause disease in plants, cause disease in humans, cause disease in animals. My own interest is in pathogens that cause disease in humans. But then, yes, you've got these different categories of, is it a virus? Is it a bacterium? Is it a parasite? Is it a fungi? And these are all very, very different types of creatures. And the reason that matters is that because of them being very different creatures, they require very different kinds of treatments, right? So if you're taking a drug to try and kill a microbe, you have to know what the microbe is and how it interacts with whether it's a human or an animal or a plant cell. And you have to know how it's different so that your treatment targets that particular microorganism and not the cells of the host that you're trying to protect. So I think we all now know a lot more about viruses in the general scheme of things. People know much more what a virus is now versus other organisms, but probably people still don't really know, what's a parasite, what's a fungi, what's a bacterium. But essentially, they're all different life forms, I guess, that differ in quite different ways. Except
viruses are not technically alive.
Well, that's true.
Which I love the idea of these sort of self-replicating robot-like things that just go in, and their sole purpose is to reproduce, and they go in, they go out, they're completely inert. They're not alive at all, and that's why I'm fascinated by them, because they subvert our host cells like little machines and make more copies of themselves and induce us to cough so that we'll spread more of these crazy robot particles. I think they're amazing because they're not alive. That's what's so fascinating about viruses.
Now, here's a tangent that we can't go on for too long because this takes hours. But
Yes.
I always think when this subject comes up, it immediately brings the question, what is life? If it is, because there are many, obviously, thoughtless creatures, living things. There are, and I know there are some people I think who would debate that but what is the cut-off point with a virus, at least, why that is not given the definition of a living thing?
One of the ways that I've described them, so I think probably how you've heard me describe them, Robin, is so to think of to think of it as hardware and software. Right. So if we think of our phone comprises the hardware, the phone itself, and then all the programs that it runs, which is the software. And so a virus is basically a piece of software that requires a piece of hardware to run, and it doesn't have its own hardware. And so the hardware is the cell that it infects, and it basically has usually something on its surface that will recognize something on the cell's surface and then use that to enter the cell. And then now it's basically loaded onto that phone, loaded onto that hardware, and then it can run. Whereas bacteria, parasites, they are the hardware and the software all put together, right? They don't need another cell. They often infect another cell. Sometimes they can stay outside of a cell or go inside of a cell and manipulate it. But they have, although there, of course, are exceptions. There are ones that have lived inside of cells for so long that they're kind of reducing all the genes that they need. So they do start to need to be with other cells. But, broadly speaking, they have hardware and software, whereas viruses are basically software. But yeah, the philosophical of quite what that means is, it's yes, another thing. And of course, bacteria can get viruses too. They're a cell, and so they get infected. And in fact, there are probably more viruses on Earth than there are bacteria, because there are so many more that can also infect bacteria as well as our own cells.
And viruses
very specific.
Viruses also get viruses, and it may go on forever like that, smaller and smaller. We just don't know.
Now, Lavanya, I wanted to ask you about this. With language generally, we hear microbes and bacteria, and I think it triggers immediately the idea that it is something bad.
Mm-hmm.
And it's that whole thing of like high in cholesterol. Oh, cholesterol's bad, and in fact, if you have no cholesterol, so can you give us some sense of the changing and understanding of actually what for ourselves, what is useful bacteria, what is not useful bacteria?
Yeah. So like while all pathogens are microorganisms, are microbes, but not all microbes are pathogenic. Many of them actually are very important for normal functioning. And I think Ed Yong's book, what was it? I have multitudes.
I contain multitudes.
I contain multitudes. Yeah, I think that was very important in bringing this conversation to the public about the fact that you have trillions of bacteria and microorganisms that are important for healthy functioning, and that's the most common example is your gut microbiome that help you digest your food and help you stave off illnesses. But you also have a respiratory microbiome, you have a skin microbiome. There's lots of useful microbes in the soil that help plants. So really a microbe isn't necessarily a bad thing. It's just a small thing, which can be good or bad, but more often than not, it's good and useful for you. But also there are some that can switch. So there are some microorganisms that are pathogenic in some contexts, but actually quite normal and won't affect you in other contexts. And one of the organisms that I study kind of falls in this category. It's called Streptococcus pneumoniae, or the pneumococcus, and it just, for many people, it just lives happily in the back of your nose and your throat, asymptomatically, which means it doesn't actually cause any symptoms. It doesn't harm you, doesn't cause any illness. But there are different versions of this bacterium that can cause very severe disease like pneumonia or even meningitis in your brain. So it's all quite complicated, but a microbe isn't a bad thing necessarily.
And also, I wanted to know about the difference between a strain and a species. So we talk about different strains. What is that?
Well, I can start, but basically they're just methods of classification, right? So strain is part of a species so that it's significantly genetically different to another strain, but strains originate from the same, what we call ancestor, from the same like original species, and they're more similar to each other genetically, now we describe them genetically, than they would be to any other species. But it's just a way to classify organisms. It's just levels of taxonomy. I don't know if you want to add anything to that, Jenny and Siouxsie.
You call it a variant, right? I mean, it's a variant. It's a slightly different version of the same species. Go ahead, Siouxsie.
Yeah. Well, I think humans are a really good example of this, right? So we're all one species, but we're all quite different depending on when our ancestors left Africa and moved around the world. And, and then as you stay in certain places, you start to change. But also as people from different ethnicities mix, then you get new variants appearing, right? So this I think is quite a good way of thinking about it. But the other thing about so you can have. So we have these species that are genetically distinct, although some of them can actually be quite similar. And there are some which you would consider. I'm thinking of some of the Clostridia that actually they're considered different species, but only perhaps because they contain a toxin or not or something. So there are ways that within a species they can change by becoming infected with viruses, which might give them extra genes like toxins or something. But that also that as they mix and match with each other, as basically as they have sex with each other, and bacteria of completely unrelated species can essentially have sex with each other, they can also change in that way. So within a species you can have lots of different strains that can actually end up being quite different. And there's a branch of microbiology that are all basically arguing about how we would even, do you call things species? Do you call things strains? I just I leave them aside and just go, we're just going to work with them.
And I think they also frequently change whether something is now different enough. This group of what we thought was strains from a particular species is now big enough and different enough that now we're going to give it its own species. So it's a way of classification and to make things easier. And they usually behave in different ways. And they can also, Siouxsie, you mentioned that they basically interact with each other and have sex with each other, but they can acquire toxins from viruses, but they can also transfer genetic material, some organisms, between each other and exchange it and to form millions of combinations that can help them acquire different characteristics. And one of the most important ones can be acquiring something that will make it resistant to antibiotics, which is very important topic to talk about. That's how strains work.
Well, I suppose that's part of, if I can ask you, Jenny, about the progress change in pathogen identification as well. What in terms of what are we able to do now? How are we able to scrutinize these things?
Well, it's made a difference. So the genomic revolution, we can now sequence things. So in my field, urinary tract infection, which nobody ever wants to talk about, it's deemed too embarrassing, but urinary tract infection is a huge problem, especially for women. Like most women's diseases, it's been historically neglected as a topic. But, diagnosis used to be and still is completely based on culture, which means you have to be able to grow the bug for the doctor to believe that you have an infection. And this is a huge problem because not all bugs grow very well in culture. And you have to have lots and lots of bugs before you'll see a signal in that culture dish. And with the advent of the sort of next generation sequencing and all these amazing technologies that have come to the fore, we can now start identifying very small amounts of bacteria and viruses and other things, not having to wait till they have to grow. And I look at you now, the TB person. If you're in a TB lab, you know that it takes months to grow some TB up, whereas I work with friendly bacteria that grow overnight. So, if your bug grows very, very, very slowly, like tuberculosis, then it's an amazing revolution that we can now sequence these things. It's like forensics. We can really dig deep into the microbiome. We can dig deep into pathogens, and when we get a new outbreak like SARS-CoV-2, we can immediately spring into action because of genomics. We were able to get the complete sequence of this virus and start working on a vaccine before we even culture the thing. So sequencing and genomics has made an absolutely massive difference to the field, and I'm so glad I'm alive for this part of the revolution, because it must have been really dreary being a microbiologist in the last century.
Would you agree with that, Siouxsie? In terms of now this chance now in the 21st century, that this way of communication, this way of being able to understand that it has been a revolution.
It has. I guess what people are seeing with COVID-19 is just how rapidly stuff can change, how even just the communication of scientific information, the fact that more people are using preprints, that, most of the time it takes a long time to do things. And then the scientists really only write, for each other. And then they usually put that in a journal that you can only access if your library subscribes to it. And so it's a very closed shop. And that's always been a bad thing for science, especially in our field where, where doctors and other medical professionals and journalists need access to our information, right? And what we're seeing now is this huge change, this massive shift in doing things much faster, doing things more in the open. It's been both a blessing and a curse. One of the things that's been quite distressing, I think, about the current situation has been what we're calling kind of academic profiteering. People who are jumping onto, and this is actually extremely true of the sequence data, because all the sequence data of all the different strains is being made available online. There are people who maybe work on genetics, but not of microorganisms, who are just sort of downloading that data and then writing papers, communicating them to very prestigious journals and getting horrendous information out there, really wrong information that they're getting very good, supposedly academic publications out of. So there are disadvantages to the way things are working, but overall, I think it's been an amazing thing. And what we're seeing also with the genomics is showing people how strains change, right? How these what we call lineages as well in viruses sort of appear. But it is amazing. I think back to, so I work on the mycobacteria as well, this sort of family of organisms that grow quite slowly. They are one of in fact, the species that we work on, they're just called non-tuberculous mycobacteria. They went from maybe, 10 species to now there's like 150, because they didn't used to be able to classify them, other than sort of some like proteins on their surface. And now we can do genomics. Mm-hmm. It's like, oh my God, there's loads of them. And one of the disadvantages of living in New Zealand is we've got very strict rules around biosecurity, to the extent that we have a law that basically says unless the organism was present in New Zealand before a certain date, it's like the 1990s or something, and by present they mean did somebody write a paper about it, it's not considered here in New Zealand, and you have to get all sorts of permissions in order to work on it. And so we've got strains of bacteria that have changed name, that have become really hard for us to actually do any work on because they're not considered in New Zealand, and yet they're actually here. And it would be exactly the same for all the bugs in our gut, right? We don't know what they all are, but they're all here, they're all living in us, but we've got this really weird quirky law that means that they're not present. That's just Anyway, so genomics has kind of changed everything in really great ways, but also in slightly complicated ways for some of us.
That almost sounds like the way our government thought that it was going to be able to beat COVID-19. Well, we just won't let those things in. We won't allow any of those microbes or pathogens. No, they won't go. Jenny, sorry, did you want to add something?
No, I'm just enjoying.
Oh, I'm sorry. I thought your hand went up in that other bit, but I think it may have just been a small amount of cramp. Lavanya, something that was kind of been talked about already, but I'd like to enlarge on a little bit, is that discussion about microbial interactions with other microbes, which was kind of hinted at a little bit. Can you give us a little bit of that story, something perhaps of an individual case to understand?
Mm-hmm. Yeah, sure. So I think we talked about this right in the beginning about how the virus, the COVID-19 SARS-CoV-2 virus, is one part of the whole disease triangle, right? So nothing exists in isolation, and microbes as well, wherever they're living, and in your body especially, they don't just live alone. There's other microbes in your respiratory tract, for example. There's human cells, there's immune cells, different types of human cells, and all of those will be metabolizing things differently and secreting different things and making the environment a very specific place. And microbes can interact with each other through so many different ways, right? They can directly secrete signaling molecules and basically talk to each other. They can affect the immune system in a way that then affects how another microorganism behaves. So if we take the example of the current pandemic, this whole idea of a predisposing condition, that certain conditions predispose you to COVID-19 is one aspect, but also that having COVID-19 will make you perhaps more susceptible to secondary infections, and these secondary bacterial or viral infections lead to a more severe or worse outcome from the disease compared to if it was just COVID-19 alone. And we're learning more about this literally every day, but there are like large-scale studies that have shown this correlation that co-infection can lead to worse outcomes. And also in the 1918 flu pandemic, they found the same thing. Like a lot of the deaths in the severe cases were because of a secondary bacterial infection and not just the initial viral infection. So this idea of like microbes living in an environment already where there are lots of things going on, but also interacting with each other and making the outcome worse is quite important to recognize, because we often will study an organism, like in very controlled conditions in the lab. And that's fair, because it's very difficult to replicate such a complex environment so we can study it accurately. But we do study microorganisms often in very isolated, controlled conditions because we want to try and understand everything about it. And then we maybe overemphasize every single like snippet of a gene signal overexpression that we get. But it's important to recognize this environment and other microorganisms that might interact with it.
Jenny, I know that you've also got some thoughts on this as well.
Yeah, I was just thinking that the opposite side of the coin is the way that the microbes can actually help us and to prevent infection. So you're talking about co-infection where two bacteria sort of synergize in the axis of evil and make you worse. But actually the opposite is true. For example, in the gut, there's lots of really friendly bacteria that are actively preventing other pathogenic bacteria from getting you. So you can find this out when you take a lot of antibiotics and you start killing off your friendly bacteria in the gut, and you'll find that you probably end up with diarrhea and maybe even a worse infection by something like C. difficile. So it's called dysbiosis. It means that there's a balance there, and when the balance is off, that's when you get sick. And we're starting to wonder now in the bladder whether this is true, because traditionally in the textbooks, urine is sterile, right? And they say if you're a Boy Scout and you cut yourself in the woods, you should rub yourself with urine because it's sterile. That's not true. Genomics has taught us that the bladder is teeming with bacteria, not surprising at all because it's open to the environment. And that we're starting to think that perhaps the bacteria in the bladder are helping to prevent urinary tract infection. And if you get the balance wrong, that's when you get sick. So there's so much we don't know about the interactions of bacteria, both good and bad.
Siouxsie, would you like to?
Yeah. This is also one of the reasons why I really hate antibacterial products. Like people always say, oh, you're a microbiologist, like you must be so clean, and you obviously don't eat food off the floor, and I'm like, no, there's a kind of 5-second rule in the house. It depends on what surface it's fallen on and what the food is. And we don't really use antibacterial products because, I know which bacteria are dangerous and what are the things that you do to prevent that. Like, you don't go putting raw meats and chicken, with, your veggies, but I'm not obsessed with this thing because I am really worried, about people who use antibacterial soaps and all these things, how you end up, disrupting all the good microbes that are there. So I think you're absolutely right that people have this very skewed idea of microbes as being all germs, and germs being a bad thing, whereas actually the vast majority of them are amazing, and we wouldn't be us without them. We're in reality, we are a bag of microbes in a human skin. We're more microbe than we are human. And so, we need to protect our little hangers-on and not try and destroy them all, because most of them are really useful for us.
Oh, I really wish I had my overflowing food bin that I've got at the moment, which has, you will be so proud of some of the things that are growing out of that at the moment. There's nothing antibacterial or anti-anything that's gone near that. The spores are ready. I want to, because we're talking on that thing, your book about antibiotic resistance. So let's move again, that bit where there are times when we think this is helpful. It's sometimes in that whole there was no woman who swallowed a fly kind of situation. We take something and we think that's going to be. So let's talk a little bit about antibiotics, because there's been a lot of talk in the last few years about antibiotic resistance and about the dangers of antibiotics. So firstly, this is just this is almost comes from me. I had a molar out the other day, which is kind of it was quite a rotten tooth, and I was given amoxicillin, which is penicillin, basically, as far as I know. And why, for instance, do I have to do the whole course? What is the reason? Even if everything ends up being fine and it turns out, but what's that reason, so that people can understand?
Yeah.
That?
Oh, so actually, this is where science is a little bit fuzzy. So, So antibiotics are basically medicines that kill bacteria. And the broad category is anti-microbials, which are drugs that kill microbes. But as we talked about at the beginning, viruses are very different from bacteria, are very different from parasites. So they have different classes of drugs. So and the real worry is, the real thing that is happening is that because these microbes evolve, all of us evolve, every cell in our, every time a cell replicates, this is true of viruses, this is true of bacteria, mistakes get made just by sheer chance. And then depending on whether that mistake is a good or a bad thing, a cell can either survive or die. And so just by sheer chance, we have got, if we take bacteria, for example, they multiply, some of them multiply quite rapidly, but they multiply to very large numbers, insane numbers. And so that means that these very small mistakes, even if they're very rare, can end up happening. And sometimes by sheer chance, these mistakes will make, if we're talking about bacteria, resistant to drugs. Mm-hmm. And then the question becomes, if the drug is present, that microbe then has an advantage. And so what's been happening with antibiotics is they are these incredible medicines that really did completely revolutionize, our health, right? Before the discovery of antibiotics these are also things made by other microbes, essentially, as they live in these communities and they're fighting with each other and they're competing with each other, they make these amazing drugs. We started using them as medicines in humans in about the 40s. And with that then came the ability to do surgery safely, all of these incredible advances in medicine, cancer chemotherapy, all of these things where you end up with a patient being vulnerable to infection. These antibiotics helped to save those patients from dying of an infection. So we've been using them huge amounts in humans, but also in animals. And that means that our world is just awash with them. Every time you take an antibiotic, the vast majority of it just goes straight out in your waste and into the environment, into wastewater treatment plants. The same with our animals, the same in the sea and in aquaculture and in horticulture. And so that means that in the environment where we know, in the soil where there are huge numbers of microbes, they are exposed to these antibiotics. And they are basically, that is selecting for these resistant organisms because they're in an environment where they have an advantage. And then because bacteria have this ability to share genetic material, they can basically just move it around. So these completely unrelated species can then pick up these resistance genes. And so you can go from, a bacteria that lives in the soil that has no relationship whatsoever to human disease becoming resistant to an antibiotic. And then through this kind of, several little steps can end up being a bacteria that lives in your nose that doesn't cause any harm to you unless it gets into your bloodstream, and then you could die because the doctor is not able to treat that bloodstream infection because it's resistant to an antibiotic. So it's this incredible thing that's been happening, which Alexander Fleming, who discovered penicillin, warned about in his Nobel Acceptance Prize. He was like, because we knew even then before they were used in humans that bacteria could become resistant to these drugs. And he said, use them really wisely. And we didn't. And so now we're in this position where for some organisms, there's basically nothing left to treat them with. And, we're in a position, in New Zealand will be the same in the UK, we're in a kind of fairly privileged position where those infections are not widespread. But there are many places in the world where they are. And then obviously because we can move around so much, although that's not quite so true anymore, perhaps in the middle of a pandemic, these organisms can move around the globe. And so it's just this kind of incredible thing where we are losing these amazing tools we have to treat infection. But it's a bit like climate change where people can't really see it happening. And so there's no real acceptance that our world is going to change. Our ability to do medicine is going to change quite significantly fairly soon. And that's a real worry because there's very little, effort being put into developing new antibiotics. And that was true before COVID, and it's going to be even worse now because the focus is entirely on COVID and viral infections.
Mm-hmm. Now, I've got a quick question for all of you. I once had some kind of gum inflammation that was so bad that I was put on whatever, and now I was put on these antibiotics where I was told these really are the ones you're not allowed to drink on. You lose 3 to 4 days if you do it. And by the way, it wasn't a gum infection. It turned out the dentist had left a large piece of metal in my mouth. That's eventually what we found out it was. But I remember that night where I was on tour with Brian Cox, and the bloke went, my wife's a doctor, of course you can drink on antibiotics. And I went, I'm on these. And he went, no, God, no, you can't. What are those antibiotics called, and why do they do that? Does anyone know? There's one in particular, which is, I think...
I know what it's called because I've been given that one as well. It's let me think about it. If someone else wants to talk about what do you know why you can't drink on them? But I think I know what this one is called.
But I found, it was fascinating. I'll give you one of the audience questions that we've had, and we come. That was a very personal. It was a fascinating thing to me. That thing which happens with infections as well, the way that they can affect the brain, the way. It's when you were talking about urine, for instance, the urinary tract infections. But this is not the question Chris has. Chris would like to know if we've known about antibiotic resistance potential since the 1940s. Could we have done anything about it, or is it just an inevitable consequence of needing to cure things?
We could have done something about it, Chris. I mean, people have been warning about this. It's been in the popular press since the 40s and 50s as well. We simply have to stop misusing them. So not taking them when you have a cold, which is caused by a virus, not spraying them all over animals. There's an interesting fact. Farmers give antibiotics to animals not to sort of protect them in advance from any infection, but because for some weird reason, they give animals a slight increase in mass. So if you treat your flock or your herd with antibiotics, they get slightly bigger. So of course, everyone in the world wants to treat their flocks with antibiotics. So they'll be slightly bigger, not really realizing that all everyone has to do is stop doing that. They'll all get slightly smaller and it won't be a competition issue. So they've been completely misused. As Siouxsie mentioned, they're impregnating the earth. The earth is awash with these chemicals. We're educating the bugs to be resistant. And I think we could have stopped doing that. We could have stopped misusing them. But it's really difficult. If you're a parent, for example, I've got a small child. When my small child is ill and I go to the doctor and I'm not sure if it's a virus or a bacteria, I'm asking that doctor for antibiotics. I can't help myself. It's sort of human nature to be worried about things like this. So there's a lot of human behavior involved. And I think the behavioral scientists will be key to helping us crack this issue of how do we wean ourselves off misusing and abusing this incredible tool that as Siouxsie says, we've squandered. When we get new antibiotics, as I'm sure we will one day, when we get new ones, we're going to have to lock them up with a key. This is called stewardship. It means when we get new antibiotics, we use them very, very sparingly and very, very carefully, and don't just throw them into the world. It's a really tough problem.
Susan.
Yeah, and one of the issues has been this diagnostics, right? Is that, and we are seeing that played out very large with the pandemic, that it is very difficult to quickly diagnose whether somebody has a bacterial infection or a viral infection. And then if they have a bacterial infection, which bacteria is it? What is the appropriate drug? I mean, this is I think is also one of the problems that we've had, that depending on how ill a person is or how demanding they are of their doctor, traditionally it's just been, just give them what we call a broad spectrum antibiotic, so something that hopefully will just hit whatever it is, rather than something very specific that will be targeted more towards the microbe that they likely have. And so this has not helped in terms of resistance. But, what we really need are these really rapid ways of when somebody turns up at their doctor's surgery or in hospital, being able to very quickly, within a few hours, say, this is what you have and this is going to be an appropriate treatment. And it's something that hasn't really been solved yet. So, what was really cool was a few years ago, I don't know whether you remember the Longitude Prize. So a few years ago, the UK kind of said, okay, well, we're going to make antibiotic resistance or antimicrobial resistance the kind of the subject of the Longitude Prize, and they made diagnostics the thing that everyone had to aim for. And I can't remember, it's a big prize that's kind of open to anyone around the world who can help solve this problem. It may well be coming to a close soon, and I don't think anybody's solved it, because it is a really, really big, difficult problem that we've always had. And the hope is that new technologies can help this along. With SARS-CoV-2, it's the use of kind of genetics and stuff, but you need material from the organism, right? And that can be really difficult when somebody turns up with hospital, they've got and possibly sepsis, so blood poisoning, but is there even a can you even get to the microbe in their blood? I mean, this is it's a really, really hard problem. And I think it gets to the heart of why we've also in human medicine misused them. That obviously doesn't excuse how they've been misused in other activities, like in agriculture.
So would it be fair to say, Lavanya, and I'll open this up to all of you, that then there's sometimes you see pieces written in the press about antibiotic resistance which suggest this is just what happens in a Darwinian world, in a Darwinian universe, but it's not necessarily. It really is about the fact that when we do get what might be considered, if we use the word, victories over how nature might attempt to debilitate us, you have to think very, very carefully, and you have to go, we use it here and we use it. So it's more down to our inability to control sometimes.
Absolutely. I don't think it's something that you just resign yourself to, that it just happens. This is just the way things work. And pathogens, well, it is true that pathogens, microorganisms will keep evolving, but to be too liberal with the use of antibiotics is definitely not the way to go. It's like elongating the life of an antibiotic for as long as you can. Basically, it's not that you can keep using one antibiotic forever, but at least try to extend its life so that it lines up with the rate of how new antibiotics are being produced, which actually isn't very fast, because like we discussed, there are many issues and many challenges in there. But definitely it's not something to just, that's how it is. Just keep taking your antibiotics. Yeah.
Siouxsie.
Well, one of the issues has also been this kind of siloing between human medicine, between agriculture, that there's a move now for what we call One Health, this kind of idea that what happens in animals, in humans, is all interrelated, right? So the fact that, you can use an antibiotic in horticulture or in agriculture, that is one that is absolutely crucial to human health, is ridiculous, right? Mm-hmm. And it's because there's been no real talk. Everybody's just been doing their little thing, and so we need to be much more joined up about how, there's very clearly reasons why you need to have drugs that work in in agriculture too, but the fact that there's no communication between the different disciplines, or at a regulatory level, the fact that people who are regulating human medicines are not the same people regulating medicines in other areas is just farcical, right? So all of that also doesn't help that people have not been there hasn't been this good interaction between the two. And here in New Zealand, so every country a few years ago was basically asked to put together an antimicrobial resistance plan, right? How is every country going to do their bit? here in New Zealand, there was a group of people who came together from both, the human side and the animal side. They put together a plan to make a plan, and it was sort of held between what we have, the Ministry of Health here, and then the Ministry of Primary Industries, which is the one that will deal with the kind of animals and stuff. And in the plan, the plan to make a plan was very clear. We have to keep these two things together. The first thing the two ministries did was split it apart. And there is now a human group and an animal group. And it's like, well, that's clearly not going to work. So
but that's what we
now,
like to hear. We need to hear that, because we see New Zealand, we see where you are as nothing more than a utopia. If we don't start finding out about your flaws, the property prices are going through the roof, and you've got to...
Yeah,
sorry, Siouxsie.
No, it's fine.
We've run out of time, but we have the live chat is now packed with people who want to know, why can you drink on some antibiotics and not others? So is there anyone here who would like to, what is it where with some antibiotics, it's not merely that it doesn't make them work, it's that it can, as we said, 3 days disappear, and then you slowly retrace your steps like a film noir. Jenny?
Yeah. I have no idea, but I want to know the answer.
Lavanya?
So the antibiotic that I think you would describe is called metronidazole. It's quite common for like oral gum infections. Now, I think we don't know why it causes this like adverse reaction with alcohol. There's obviously some sort of reaction that happens, but what I suspect, an educated guess, is when they, every drug, every vaccine has to go through stringent safety efficacy trials. So probably when they were going through these trials, they found that some people who were drinking alcohol and taking the antibiotic had some very serious side effects. And so they said, you know what, just don't do it. Otherwise it works perfectly. So my guess is they observed this probably at like the trial stage and decided that it's serious enough and it happened in like a large enough proportion of their their test subjects that they decided just to say don't do it. But we don't know, actually. I did a quick search of why, but I don't think we know exactly what happens.
Siouxsie, have you anything to add on that?
It could be that, I guess, drugs are chemicals, right? And sometimes when they act, they get turned into other chemicals. Mm-hmm. And so it could be either something that is alcohol impacts on, either one of the byproducts or this chemical to make something that's quite noxious to us. It could also be that, in some people, maybe either their gut microbes or something metabolize alcohol in a way that produces a byproduct again that can work kind of in a bad way with either this drug or byproducts of this drug. So these things happen. And I think another one is grapefruit juice. There are drugs that you can't drink grapefruit juice with. And so it'll be about sometimes just these kind of what we call, metabolites or byproducts that are made that then interact in a terrible way. But it's kind of fascinating and amazing.
So there we are. Much like consciousness, antibiotic insanity still remains a mystery. It is yet another of the hard problems. Thank you so much to Jenny, to Lavanya, to Siouxsie for joining us. As I said, all of these episodes are available. You can also get audio versions as well if you're out jogging or something like that. And I would also like to thank Aziz Genetic Society and the Milner Centre for Evolution at the University of Bath, and also to our producer, Trent Burton. We're back in a fortnight for another live episode. And I will remind you again, regular viewers, that we are going to do 2 episodes for those ones where suddenly everyone sent their questions at 9:15. We are going to do some follow-ups and we are going to do some Q&As with those. But we're back in a fortnight, and I'm not sure. Do you know what? We're going to do another expert panel on COVID-19 as well. So tweet us any questions you want. And we are hoping, I think, in the next few weeks, we're going to try and do a few short kind of expert films explaining some of the things that people keep bringing up, some of the kind of conspiracy theories and some of the things about certain health advice which people aren't taking, again very often from a kind of conspiracy mindset, or I would say a misled libertarian point of view. We'll be dealing with that as well. So thank you very much, everyone. We'll see you at the next Genetic Shambles. Bye bye.